And yes, so the trial, of course, as you have heard already, probably it's designed to define what are the existing remodeling which cause the incidence of atrial fibrillation in a restricted cohort of patients at risk. So in the trial, the PREDICT-AF trial, there are 150 patients enrolled. And these patients, of course, are screened for specific risk factors and exclusion criteria. And they all have to go elective surgery for coronary artery bypass grafting or valvular replacement. And during these procedures, there is an excision of part of the left atrial appendage and some peripheral blood collection from which the authors of the trial want to estimate what are the relevant markers which are discriminative for incident AF. After biobanking, the discovery happens in 4 phases. There is an untargeted phase in which 22 patients are sampled, 5 with incident AF, we will see later, for RNA sequencing, and which will determine which set of genes and which pathways are correlated with future incidence of AF. Then we have selected from this RNA sequencing, we have a selected number of genes which are confirmed in terms of quantitative expression via qPCR or real-time PCR. And there is also immunohistology to confirm that there is indeed, we'll see later, that there is extracellular matrix deposition or activation of mesenchymal cells with the preservation of microvascularization. And in order to compare and to estimate which relevant biomarkers in the tissue is possible to observe also in the peripheral blood, there is on all the patients, there is detection of selected proteins via ELISA. In the trial, there are 2 follow-ups followed. There are 2 outcomes followed. The primary is incident AF with which is, of course, clinically determined as atrial tachyarrhythmia on ECG for 30 seconds of continuous recording. And this has a cutoff day more than 50 days after surgery and within 2 years. There is the maximum duration of the follow-up. And the secondary outcome of the trial is postoperative atrial fibrillation, which means less than 50 days post-surgery, that then it's not, there is not followed by an incident AF after. Okay. Here you can see the inclusion criteria of the patients. So it's very important to view that this is a cohort of patients which is actually at risk of developing So aside from the elective sternotomy, they had a CHA2DS2-VASc score equal to at least. But in the so they are recommended in the so they are recommended time of the first screening. The time of the first screening, they are in Simon Street, and the exclusion criteria are quite stringent. But the exclusion criteria are quite stringent. All the possible comorbidities, can explain atrial fibrillation from other sources. Atrial fibrillation. The patient cohort is during the follow-up. Only a few patients were lost. And we can see the initial results of the study in this slide. So first the authors try to determine whether can predict the incidence of can predict the incidence within 2 years. And they found out that 2 years, none of them does. And they found out that actually none of them does. Even the age is just close to significance. They determined that none of the clinical risk factors can predict the secondary outcome. Some clinical risk factors, some clinical risk factors can predict it. In particular, the absence of concomitance, and valvular replacement bypass. As you can see here, it's summarized. Number of patients who completed the follow-up, presented with incident AF. The majority of them presented also.
These samples were taken during surgery, and it is possible that in the post-operative phase of surgery there is a development of inflammatory response or whatever. How do they account for this? I mean, they measure it at the wrong moment, don't you think so?
No, I didn't see any inflammatory follow-up in the paper. No, I don't have it in my notes. The patients were monitored during post-op intense in the ICU, for sure. But it wasn't reported. It wasn't reported any particular anti-inflammatory regime.
No,
as far as I remember.
But of course, you won't find any inflammatory markers if you measure before the.
No, of course, no. That's true.
Okay, thanks.
That's true. But yes, the authors also discuss the 50 days cutoff for postoperative atrial fibrillation versus incidence AF, and they show that 30 days the results are basically the same. So they kept with 50 days cutoff days for postoperative atrial fibrillation. But it's true that this is not reported whether there was an inflammatory response due to surgery. For sure, there was some remodeling due to surgery. So continuing, let's look at the RNA sequencing, which is very interesting from my side. So 22 patients were selected for, so the samples, especially I'm talking about the left atrial appendage, half of it is frozen in liquid nitrogen for biomolecular assays, and half of it is used for histological analysis. So part of the frozen samples are used for RNA sequencing, and 22 patients, of which 5 had incident AF and 17 were did not develop it, so were used as control. And as you can see here from the Volcano plot, there are here on the left, there are several genes which were found differentially transcripted, sorry, and by gene analysis, several pathways resulted upregulated and several downregulated. So as you can see, all the upregulated pathways are somehow connected with handling of the extracellular matrix. So deposition of proteoglycans, glycoproteins, integrin surface interaction, a very interesting epithelial to mesenchymal transition, which is suspected to be one of the way to which fibrosis happens from neodifferentiation from the endothelium to fibroblasts. What is really interesting for the side of the cardiometabolism is that all the downregulated pathways are somehow connected to cardiac metabolism. In particular, creation of new fat tissue and handling of adipose tissue, and mitochondrial respiration is completely downregulated, and even mitochondrial translation is downregulated somehow. So this is not a focus of the paper, but I would like to go back to it during the discussion, because it's really interesting that some of the downregulated pathways are actually in logfold higher than the upregulated ones. And the number in the supplementary materials, in the number of upregulated pathways and the downregulated pathways are roughly the same, are 84 versus 89. So this is a very interesting finding from the study, and it's kind of I don't want to say it's briefly explained as in the paper as a striking evidence, because from the animal models we think that intense energy needs and therefore movement between shifting between fatty acid beta oxidation to glucose utilization is due to the progression of AF. But actually here it seems that the metabolic deregulation precedes atrial fibrillation. Anyway, going back to the focus of the paper, the authors of the paper decide to focus on the upregulated pathway. It's very interesting. The two main upregulated genes are collagen A2A. So the gene which expresses collagen strand fundamental for the basement membrane, and fucosyltransferase 4. Which catalyzes glycosylation of proteins. This is something that we also observe in our study on animals, that because you are using more glucose for energy production, then you have more glycated proteins. But stemming from the RNA sequencing finding, the authors, as I was saying before, selected the 26 genes based on the finding of the RNA sequencing and the historical relevance of the genes, of certain genes, especially the COL1A1, that are known to participate into AF progression. And they observed with qPCR what are the significantly differentially expressed ones. So, interestingly, there is only one gene, a regulatory subunit of the sodium channel, that is overexpressed. So we tend to assume that electrical remodeling or structural remodeling both precede somehow the occurrence of AF, but it doesn't look like it from here. I mean, there is only one gene overexpressed. What is really overexpressed is genes involved in the handling of the extracellular matrix, especially in the production of extracellular matrix. So we found fibrillar collagen 138. Well, this is not all fibrillar, biglycan, thrombospondin, and especially tenascin C. We will see later it will become more important, and endothelin 1, which is also an activating agent of fibroblasts. And there is also fucosyltransferase here, but it's not significant in qPCR. So based on that, based on these genes found by confirmed by qPCR, the authors wonder whether these genes are overexpressed genes are. It's also possible to detect a change in the circulating protein in the plasma of the patients. So what they can find is that only tenascin C is significantly increased at the circulating level of the plasma of the patients, and this is actually visible in this figure when the correlation between the tissue tenascin and the plasma tenascin is positively correlated and significantly correlated. I have not clear what this picture, the B right, stands for. It's written in the legend that they eliminated this extreme value, and the correlation seems lost between the tenascin expression in the tissue and the plasma levels. But this is not mentioned in the paper. So yes, it's a pity because tenascin is proved to be changed in myocardial infarction, and it's determinant for the contact between the cardiomyocyte and the basement membrane. So it's a pity, but in the paper this is an obscure point. It looks like without this extreme value, the correlation is lost. So collagen 8 in the tissue and in the plasma is not, as you can see using all the patients, is not correlated, and it's of course the p-value is also not significant. But if we take only the above average number of patients, then the authors claim and they show that a correlation between plasma levels and tissue expression exists and is significant. Of course, this can mean that there needs to be a significant expression of collagen 8 to have it circulating in plasma in excess enough to be detected by ELISA. They say that if we consider all the patients, there is no correlation. Okay. If we take the above average tissue expression, and we plot them versus the plasma levels, then a correlation appears. And while this might be mathematically artificial, I mean, at the level of the body, I can understand that probably we are like below this threshold. We are under a relevant level of circulating collagen 8A to be correlated with anything in the body.
Mm. But if you do the correlation on the left part, you will find a negative correlation.
That's why I said that it might be mathematically artificial. So these are the findings, and unfortunately they are disappointing because it looks like there is no plasma correlation with the tissue markers, and this is a pity, obviously, because we would like to see it in the without surgery. So the latest part of the paper focuses on the selection of a gene cluster, if it's possible to be identified, to estimate better than with clinical risk factors whether a patient will have incident AF. And with gene cluster analysis, the author have found this set of genes. So bioglycan expression, collagen 8, endothelin 1, and downregulation of the potassium channel KCN, the gene KCNJ2. And they proved by doing C-statistics that if they so C-statistics is this number is the area under the ROC curve, and of course it gives you the sensitivity versus specificity of your dichotomous determination. The patient based on this parameter will not have incident AF or will have incident AF. And they found out that if we include the left atrial volume index on age or canonically associated circulating markers and T-proBNP, This have a little specificity, but if we include collagen 8 and tenascin, this starts to go up. And if we include all the genes in the cluster identified, then we have the higher area under the curve. Of course, it's nice to see. It's also disappointing that we don't have plasma biomarkers included in this, but these are just genes. So it means that we would have to take the tissue and look at the gene expression, and myocardial biopsies are not easy to do. They're also not ethical most of the times. The last part that is presented by the authors is the histological changes that can be found in the atria of patients which will develop AF. And as we can see, there is inside the myocardium, they could not see with Pygosirius red and with Vimentin and CD31 staining, they could not see any significant change in interstitial collagen between the group that developed AF and the group that did not. The total collagen level was similar, but when they went to analyze the epicardial fibrotic area and the epicardial mesenchymal cells relative quantity to cardiomyocytes, they have observed that it looks the patients that will develop AF appear to have an increased fibrotic area on the epicardium and an increased number of mesenchymal cells, which are probably fibroblasts, but not more interstitial mesenchymal stem cells, which suggests that the activation of the structural remodeling starts. It's region dependent and probably starts from the epicardium, which is not surprising because the debate on epicardial activation is still active, especially with fibrofatty infiltration and fibroblast proliferation. And this is there is also a figure in the supplementary materials when they have stained the microvasculature, and they show that there is no difference between the two groups in terms of microvessels density. So there is no microischemic region that might explain or that might suggest another pathway for the incidence of AF in these patients. And this is the end of my presentation. Thank you very much. Sorry for the technical issues.
That was a very good presentation. You already mentioned it, the influence of the metabolism pathways is quite interesting because it could point to a causative factor in the genesis of AF. On the other hand, we know that in heart failure, changes like this also occur. The change from lipid metabolism to glucose metabolism, for instance. Do you think that this is related to AF or maybe congestive heart failure or other diseases?
It looks like to me this belongs to a bigger debate whether AF can be classified as a symptom of something or a disease per se. I will not share my opinion on this because it's still forming mainly. I think personally that I was looking for the NIA class of these patients before, but I don't see it. There is only one patient with congestive heart failure diagnosed. Of course, it's most of these patients underwent also valvular surgery. Yeah. So I don't feel 100% confidence to say that these metabolic changes were not secondary to some valvular disease, yeah, which is it's a typical comorbidity of atrial fibrillation. But I believe that also from our study there is a significant susceptibility that is given by metabolic downregulation in the atrium. So this might be a little bit blurred with an existing, at least valvular or ventricular problem, because these patients went bypass grafting. So
yeah, their ejection fraction is not very low.
Yeah, exactly. It's not very
okay, I suppose.
So I am relatively confident that this is at least one manifestation of atrial fibrillation that presents initial downregulation of metabolic pathways, which is in comorbidity with valvular diseases. But it is striking for me to see in patients these things that we can observe there is also a lot of literature on rapid atrial pacing animals that have metabolic shifts, and especially I was expecting mostly a shift in fatty acid metabolism, but here I see mostly the 3 TCA cycles. So the basic function of mitochondrial role is depressed, and this is also connected, if you see here, with peroxisomal transport. So this can suggest that it's concomitant with a state of oxidative stress inside the tissue. But these are very interesting and subtle findings, and you can see here the cardiac muscle contraction. It's also downregulated, and this is known that when there is mitochondrial degeneration, there is consequent myolysis. So there is also problems at the level of the contractile machinery. So this is actually my question, is which I would like to ask to the author actually. Why did they debate these metabolic changes in 4 lines? Basically, in the online version is 4 lines, or in a short paragraph of the paper. This is really interesting.
Well, we can ask them.
But we can ask them. But as you said, Ruben, I think it's because the metabolism it's more difficult to isolate as a single chamber-specific phenomenon.
Yeah, I agree. Yeah, but if you have a question to the author, there's nothing against it if you write them a mail, because
of course
it's very interesting. And of course, we can act as intermediate, but
I would love that.
I suggest that you write a mail to them and ask the question.
Sorry, so one thing that is not in my presentation, the results that we have seen with the qPCR, the RNA sequencing. So these are all related to the incident AF cohort, but the authors also show in the supplements that in the postoperative atrial fibrillation cohort, these differences do not exist. So this is why they don't talk about them in the main paper.
How are you going to follow up on the metabolism?
Well, we cannot do this. We didn't have prepared any study like this yet. But like with Pierre Dos Santos, for example, we are thinking seriously how to isolate a specific cohort which will not present significant metabolic confounders. I must say that it's not reassuring that or comforting that the initial exclusion criteria filled 1 page. Basically, we needed a healthy patient with atrial fibrillation. No, we are working hard, especially compared with this paper, to find out what what is the right cohort? We have we have some things to avoid, of course. As I said, metabolic syndrome, at least 2 out of 3. The conditions, dyslipidemia, this kind of stuff. Also, we have to be careful on glycated hemoglobins because the patient might not be diabetic, but might be in the range. Insulin resistant is a huge problem for glucose metabolism and fatty acids regulation. So we have to be careful. There is a lot of literature to go through.
Yeah.
But this is the main problem now that we are facing. Looking at this paper, it's raising a question on what is representative, basically, in the patients, among the patients. These downregulations are really interesting, and we hope to find a good match in our animal models.
We have to stop. Still, we had an interesting discussion in the limited time that was there. A nice paper.
Thank you.
Then I'll see you all at December 6th.